oogonia, and then differentiate to the primary oocyte, secondary oocyte, and the rest of them. What about the sperm? This PGC changing to spermatogonia, something like oogonia. It's changing to spermatogonia, and in the process of spermatogenesis, it wants to change or transform into spermatozoa. The spermatozoa is the things that a man needs for fertilization. Okay? Look at here. We have the germ cells here. These ones. Primordial germ cells. And between them, we have a cell, we call it Sertoli cells. Yes, supporting from different aspects. Nutrition and phagocytosis, I will tell you how it can happen. So the Sertoli cells is the supporting cells. Where is the origination of these Sertoli cells? How the Sertoli cells is made? Where is the origination?
Yes?
No. Similar to follicular cells in women. I told you we had primary oocyte, and also I told you we have this surrounding layer. I told you we called it follicular cells. I told you these cells coming from the epithelium of the ovary. And here, this one, the Sertoli cells, this one, the Sertoli cells, also coming from the surface epithelium. But in the male, we don't have ovary, we have testes. So the origination of these Sertoli cells, which wants to support, and these cells, follicular cells in women, is the same. Both of them are from epithelium. One of them from epithelium of the ovary, another one coming from epithelium of the testis. Another name for the Sertoli cells is sustentacular cells. And it wants to support. You are seeing the location of the primordial germ cells and the Sertoli cells. Before the puberty, in the testes, we have the cord. We have cord. Imagine the cord. Before the puberty, this cord is starting to be a tubule. We call it something, for example, something like a tube. Something which have a lumen. Okay? So, this sex cord needs to have a lumen to become the seminiferous tubules. And in the puberty, these primordial germ cells started to change to spermatogonial cells. Similar to women, these spermatogonial cells starting to mitotic divisions. These spermatogonial cells, by means of the mitotic division, changing to spermatogonia type A. Then again, mitotic division, and making spermatogonia type B. After the spermatogonia type B, it changed to primary spermatocyte. Do you remember, in the female we have primary oocyte. Here we have primary spermatocyte. Both of them starting the meiosis. Before these two, primary oocyte and spermatocyte, we have just mitotic divisions, increasing the number. But after that, we will go to meiosis process. And you are seeing here, this is a section of seminiferous tubules. It means that we are seeing all of the cells. At the base, we have spermatogonia cells. You are seeing spermatogonia. And I told you, between them we have this supporting cell, Sertoli cells. They are starting to divisions, and after that we have primary spermatocyte. And you are seeing that again, this primary spermatocyte divide, and making this spermatid. And after that, they are going to the lumen of the seminiferous tubule, and then go to the epididymis and store for fertilization. Okay? So this is the process of spermatogenesis. You are seeing it is started by type A, then mitotic division, type B, and after the type B, it goes to primary spermatocyte. Then go to secondary spermatocyte, and at the end of the meiosis number 2, we have a spermatid. But at the end of the meiosis 2, the spermatid is not ready for fertilization. It needs another process. We called it spermiogenesis, I will tell you. And if you look here, all of these cells connecting to each other. Look at here. All of these cells connecting to each other. Why these are connecting to each other? I told you when two cells wants to be separated, the cytoplasm needs to be divided. But until this part, the cytokinesis is not completed. It is incomplete. When it will become it will be completed, the cells being separate. When it will happen when the spermiogenesis is completed and we have the ready sperm. Before that, they are connecting to each other because the cytokinesis is incompleted. And you are seeing, again, I told you about the Sertoli cells. What was the function? Support. Your friend said that for sending the nutrition. And also for protect these germ cells. Support and protect these cells. These are very important for us. These are wants to be a sperm. So they must be protected. They must give, for example, the nutrition from the Sertoli cells. And what happened in spermatogenesis? I told you in the female, LH was very important. Again, in the spermatogenesis is also LH is very important. LH and FSH, both of them are very important. Look at the picture. Here we have the hypothalamus in the brain. In one part of the brain we call it diencephal, around the third ventricle in our brain. This hypothalamus, you don't have any function in your body that the hypothalamus doesn't involve. It's very, 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 very important things in our brain. I told you, you don't have any function in your body without involving the hypothalamus. So the hypothalamus secreting this hormone, gonadotropin releasing hormone. It means that it is a hormone which causes that the gonadotropin is starting to release. Release from which part? From the pituitary gland, which again is located in the skull. After that, what happened? The LH is secreted. And also the FSH is secreted. The LH and FSH going and affecting on the cells. One cells here we have Leydig cells, and also we have Sertoli cells. How it affects? This LH binds and attaches to the Leydig cells, these cells. Why attach? To make what? To make testosterone. So after that the testosterone is made, and then this testosterone coming and attaching to Sertoli cells. So from the hypothalamus, pituitary gland, secreting the LH and FSH, they are coming, LH attached to the Leydig cells and causing to secreting the testosterone, and this testosterone affects the Sertoli cells. And also we have the FSH. Again, FSH coming and affecting the Sertoli cells. So both of them, LH and FSH, involving in spermatogenesis. You are seeing it's come and attached to the Sertoli cells to make what? To make a fluid and to make one androgen receptors. Because the sperm is not enough for fertilization. It needs something and other things, and they are making by means of the Sertoli cells and also coming from other parts. So you understand that the LH and FSH are very important in spermatogenesis by affecting the Leydig cells to make testosterone, and then the testosterone affects the Sertoli cells, and also this FSH also affects the Sertoli cells to make androgen and also the fluid production. Do you have a question? Question? No? The next part is spermiogenesis. I told you the spermatogenesis is not enough for making a ready sperm. We need another thing, we call it spermiogenesis. What happened in spermiogenesis? We have several things. Formation of the acrosome. Yes? Who said? Tell me.
The growth of the sperm.
Yes, it helps to not growing the sperm, making it ready. Do you remember, for example, I told you, here is the oocyte, here, and I told you, here we have the zona placida. The sperm wants to penetrate and going inside the oocyte. Yes, it needs the acrosome to do what? To pass from the zona placida. So the sperm, the early spermatid, doesn't have any acrosome, and it cannot penetrate the oocyte, penetrate the zona placida layer of the oocyte. It needs acrosome. So formation of the acrosome. How the acrosome is helping to penetrate? Because it has several. Yes. Because of the enzymes, it's being ready for penetrating. And this sperm also needed tail and middle piece. Why it needed middle piece? Because yes,
control the movement.
to help the movement. How?
Control.
Making ATP?
Yes. By means of the making energy. This sperm wants to go, wants to move. Okay? So it needs energy. The energy or the ATP is made in the middle piece. And it needs to swing. So it needs tail. So what happened in spermiogenesis? At first, the acrosome is made. And after that, this nucleus is being condensed. So you are seeing that this acrosome is covering this nucleus. And at the next stage, you are seeing that the middle piece and the tail is made. And in the next stage, the Sertoli cells coming to do what?
The rest of the cytoplasm will be like residual bodies which will be absorbed by the Sertoli cells.
Completely true. So the Sertoli cells come and clear this cytoplasm. Phagocytose this cytoplasm and making this sperm. It's ready for fertilization. But in the women, when the ovum is made, it's ready for fertilization. It doesn't need any another process. It's also be changing into the time. I will tell you, because of the sperm is coming, one thing happened in the ovum. But it doesn't need any specific or any extra process. But in the male, we have this spermiogenesis process, which includes formation of the acrosome, condensation of the nucleus, making the middle piece and the tail for making the energy and also for swimming and movement. And after that, this cytoplasm needs to be removed. And after that, we have the ready spermatid. This process, making the sperm, taking time, 74 days. And each day, 300 million sperm is made. And after that, I told you, at first we have cord, sex cords. It doesn't have any lumen. But during the puberty, this cord changing to tube. Imagine that, for example, at the first time, it's something like this, the cord. But we need something to move. And this is the tube. So these are, you are seeing that. The spermatogonia changing to spermatocyte, the spermatocyte doing the meiosis, making the round spermatid before the spermiogenesis, and then elongating spermatid after the spermiogenesis, and then this spermatozoa coming to the lumen. What happened after that? It's going to this part of the testis, epididymis. Before this part, before going to epididymis, it cannot move very well. It doesn't have ability to move. But in the epididymis, it can be mobile. It can be moved. Okay? So that you are seeing that it's obtained full motility and or the movement in epididymal parts of testes.
Doctor,
Yes?
we said that our cells are not living.
Not living.
Yes, I mean by living, not individual living cells. So, okay, how can sperms turn to living cells?
How can the sperm change to?
Living, individual living cells, so that they can move by their own. What kind of intelligence, what kind of thing can give them this?
It has it obtained the ability. I told you at the first time. I told you at the previous slide. The sperm is making. The sperm is made. It cannot move. So it goes to epididymis. It obtained the ability to move. Then it goes to another part. You will see in anatomy. It gets the ability, for example, to being alive. Then it gets the ability, for example, to for ejaculation. Okay. The earliest spermatid is not enough for fertilization. It needs to be, for example, it is starting spermatogonia, starting, then primary ovocyte, secondary ovocyte, early spermatid, late spermatid. Then another thing happened. Imagine that the snow is coming. It's stored at the mountain. Lots of the snow coming to this snow and making a store. We need this store for fertilization. Prostate sending something. We have lots of the gland, bulbourethral gland, which secrete something and add to the sperm. By means of adding several structures, we have the ready sperm. For example, when you are cooking something, you have raw things, raw meat, raw nuts, for example, raw rice. You started to make things, and at the end you have a food. In the sperm is also similar. At first you have just unmobile sperm. Another thing come, and also the brain helps. by means of releasing the hormones.
I'm asking if
You can rest for 5 minutes and you come class. Don't waste the time on other people.
they have the ability to